Photosynthesis is nature's most sophisticated molecular heist. It is a high-stakes relay that powers nearly every living thing on Earth. It begins when a photon of light strikes the chlorophyll molecule. This single impact is the spark of life. The photon energy destabilizes an electron, ejecting it from its ground state. This electron is now a vehicle for captured solar power. This electron is passed through a relay of proteins in the membrane, performing the heavy lifting that drives the production of biological energy. This molecular heist captures fleeting sunlight, locking it into the stable chemical bonds that sustain our entire biosphere. Within the chloroplast, we find the thylakoid, a specialized membrane system acting as a microscopic energy turbine. Light harvesting complexes are organized precisely across these membranes. They function as antennas, capturing solar radiation. Photons strike these pigment clusters, achieving nearly 95% efficiency through quantum coherence, effectively trapping light energy. As high-energy electrons navigate the electron transport chain, they release energy that drives the proton pump mechanism. This energy allows specific proteins to shuttle protons or H+ ions from the stroma into the thylakoid lumen. This massive accumulation of protons creates an electrochemical gradient, effectively charging the membrane like a biological battery. The ATP synthase is a molecular motor functioning much like a hydroelectric turbine. A high concentration of protons in the thylakoid lumen creates an electrochemical gradient. As protons flow back into the stroma through the enzyme, their kinetic energy forces the internal stalk to rotate at incredible speeds. This rotational energy provides the force necessary to catalyze the bonding of a phosphate group to ADP, forging ATP, the primary energy currency for cellular work. In the light-dependent reactions of photosynthesis, the production of ATP is only half the story. We also generate NADPH, a vital molecule that acts as a mobile carrier for high-energy electrons. NADP+ accepts a pair of high-energy electrons and a proton, effectively charging the molecule with reducing power. Watch as the electron carrier molecule is transformed. This NADPH molecule is now primed. It carries the necessary reducing power into the stroma, where it will donate these electrons to drive the carbon fixation processes of the Calvin cycle. Without NADPH, the plant could not successfully convert inorganic carbon into the sugars that sustain nearly all life on Earth. In the stroma of the chloroplast, the Calvin cycle transforms inorganic carbon dioxide into organic sugar precursors. Atmospheric carbon dioxide enters the cycle, acting as the foundation for the sugar molecule. ATP and NADPH, generated in the light reactions, provide the necessary chemical energy and reducing power to drive the fixation process. Through a series of complex reactions, carbon atoms are rearranged, ultimately producing G3P, the essential precursor to glucose. This cycle locks sunlight into physical chemical bonds, sustaining life across the planet. RuBisCO is the engine of the Calvin cycle. It captures inorganic carbon dioxide and fixes it into organic molecules. As the most abundant enzyme on Earth, it acts as the vital gateway for carbon to enter the biosphere. In the Calvin cycle, inorganic carbon dioxide is captured and transformed into G3P, a 3-carbon sugar that serves as the foundation for all plant life. First, carbon dioxide is fixed to a 5-carbon molecule, creating a transient intermediate that immediately splits into 2 3-carbon PGA molecules. Energy from ATP and reducing power from NADPH are then applied, converting these PGA molecules into G3P. This G3P molecule is the essential building block. From here, the plant constructs glucose, starch, and cellulose, effectively turning sunlight into physical matter. The G3P molecule is the primary output of carbon fixation. It is the architectural starting point for the biosphere's energy. These molecules are synthesized into glucose and complex starches. This is how the cell locks solar energy into stable chemical bonds. This stored glucose provides the caloric foundation for the entire food web. From primary producers to the highest apex predators, all life depends on this moment of energy capture. Without this specific process of carbon fixation, the movement, heartbeat, and thought of every animal on Earth would simply not exist. RuBisCO is the engine of life, but it has a famous flaw: photorespiration. In this process, the enzyme accidentally grabs oxygen instead of carbon dioxide. By 2026, researchers successfully engineered a hyper-efficient variant. This synthetic enzyme uses quantum structural precision to ignore oxygen molecules entirely. Watch as the new variant maintains its focus. Even with oxygen present, it exclusively binds to CO2, streamlining the production of the glucose precursor, G3P. This breakthrough minimizes energy waste and allows plants to convert solar energy into chemical fuel at unprecedented speeds, significantly increasing global crop yields. We are moving beyond biology. Synthetic photosynthesis uses man-made catalysts that mimic the architecture of photosystem II. These artificial leaves are integrated into building facades, turning urban skyscrapers into active, carbon-capturing surfaces. By capturing sunlight, these catalysts split water and fix carbon dioxide directly into liquid methanol. This turns our cities into solar-to-fuel factories, creating a path to true carbon-negative power. We are currently witnessing a shift in architecture, where buildings are no longer just shelters, but active participants in climate restoration. By coating skyscrapers in biomimetic skins, these structures function like artificial leaves, performing direct air capture of carbon dioxide. Using integrated catalysts, captured carbon is converted into liquid methanol using solar energy, effectively turning skyscrapers into carbon-negative power plants. This biomimetic approach ensures that our urban centers contribute to, rather than consume, the world's carbon budget. Natural photosynthesis operates within a thermodynamic limit of roughly 5% solar to biomass efficiency. It is the baseline, but nature has constraints. By mid-2026, synthetic systems achieved a historic 12% solar to fuel efficiency. This milestone effectively doubles the natural limit, marking a leap in energy capture. This advancement offers a scalable path to food and fuel production that is largely independent of traditional land use constraints. We are moving toward a future where efficiency defines sustainability. We are now engineering hybrid systems where bacteria are augmented with silicon nanowire arrays. These biosynthetic machines perform photosynthesis at 10 times the efficiency of natural vegetation. Every year, the global photosynthetic process fixes approximately 120 billion metric tons of carbon from our atmosphere. This constant breathing of the planet acts as an atmospheric regulator, locking carbon into stable chemical bonds that sustain all biological life. Without this massive biological engine, the atmospheric composition would fail to support the complex ecosystems we rely on today. Consider this remarkable fact. 99% of all your daily movement and thought is derived from energy that spent less than 48 hours as a solar photon before becoming a chemical bond within a plant cell. Plants act as the primary interface for our biosphere. They capture solar flux and store it as chemical potential. This energy transitions from a raw photon into a stable chemical bond, essentially a compressed package of solar power. From the plant cell to your own mitochondria, this relay happens on a remarkably short timescale. We are not living off ancient reserves, but off the real-time heartbeat of the sun. We are powered by the immediate, real-time capture of solar energy, bridging the gap between stars and life itself. For a century, humanity has powered its growth by tapping into the Carboniferous paradigm, burning the saved light of ancient forests buried deep beneath our feet. This era of reliance on finite, buried resources is ending. We are transitioning to a new architecture of life, harvesting the real-time energy flux. By mimicking natural photosynthesis, we can capture photons as they arrive, turning our cities into factories of carbon-neutral fuel. This fundamental shift allows our species to power its future using the steady, inexhaustible rhythm of the sun, moving beyond the ancient carbon trap. Nature's molecular relay is a marvel, but it is fundamentally limited by evolutionary constraints. Biological systems evolved for survival and robustness, not necessarily for peak energy throughput. The primary bottleneck is the enzyme RuBisCO, which evolved in an atmosphere with vastly different oxygen concentrations. It is famously inefficient, often grabbing oxygen when it should grab carbon. We are now rewriting this code. By bypassing evolutionary trade-offs, we are transforming photosynthesis from a limited biological curiosity into a high-output energy machine. By streamlining electron transport and refining catalytic enzymes, synthetic pathways achieve energy outputs far beyond what a wild plant achieves in nature. Photosynthesis is more than just a biological curiosity. It is the ultimate blueprint for the future. From the quantum precision of a single leaf to the vast scale of global carbon sequestration, we are mastering the light that powers our existence. We are bridging the gap between natural biological efficiency and human innovation. By mimicking these molecular pathways, we are transforming our infrastructure into carbon-negative power plants. The transition to a solar-powered species is not just a technological upgrade, it is a fundamental shift in how we power our civilization. We are moving from harvesting ancient, buried light to harvesting real-time energy flux.